Hello, today we are going to discuss Trypanosoma brucei, which is the causative agent of sleeping sickness. Trypanosoma brucei belongs to the kingdom Protista, phylum Euglenozoa, class Kinetoplastia, order Trypanosomatidae, family Trypanosomatidae, and Trypanosoma brucei being the genus and species. Moving on to the life cycle of Trypanosoma brucei, we see that it is transported via the tsetse fly, which acts as a vector. Humans are the primary host, and cattle or other wild ungulates act as the reservoir host. Here we have a microscopic view of Trypanosoma with labeled cellular structures and an image of Trypanosoma brucei in human blood under the electron micrograph. Trypanosoma brucei employs various strategies to modulate both innate and adaptive immune responses from the host. Antigenic variation, driven by the variant surface glycoprotein, or VSG, allows the parasite to continuously change its surface, avoiding antibody-mediated clearance. The tight packing of dimers also creates a protective coat around the cell and flagellum. In addition, Trypanosoma brucei employs a serum resistance associated protein to cause disease in its host. It also uses glycoprotein that is unique to Trypanosoma brucei called TB gambiense specific glycoprotein that codes for a shortened sequence of the VSG, which contributes to the surface variation. If we were to inhibit VSG, the parasite would become susceptible to APOL1 toxicity from host cells. APOL1 disrupts ionic homeostasis, leading to trypanosome death. Inhibiting VSG would benefit the host while harming the parasite. If I were to explain this to a patient, I would explain that the parasite uses a vector, or insect in this case, to infect the primary host. Once inside, it must hide from the human immune system, achieved through changes in the surface protein, the VSG. This surface protein is also important in creating a protective layer around the parasite. Inhibiting VSG renders the parasite more susceptible, leading to host-produced APOL1 to enter the cell membrane and cause lysis. Here is a diagram that shows the role of APOL1 and its role in the immune system. Treatment depends on the weight, age, and severity of infection of the host. Stage 1 of treatment involves pentamidine or fexinidazole. Stage 2 of treatment typically employs a combination of nifurtimox and eflornithine, or NECT for short, or fedinazole if it wasn't used in stage 1. The figure shows the drugs that are recommended for treatment. Let's look at the mechanism of these drugs. Pentamidine interacts with Trypanosoma kinetoplast DNA by cross-linking adenines in the AT-rich portion of the DNA. This leads to suppressing type 2 topoisomerase in the mitochondria, which leaves the mitochondrial genome fragmented and unreadable. This drug also inhibits polyamine synthesis due to decreased activity of ornithine. It also inhibits RNA polymerase synthesis and nucleic acids and proteins. NECT involves eflornithine, which is an irreversible inhibitor of ornithine decarboxylase, which acts as the primary catalyst of ornithine into polyamines. This inhibits cell division and differentiation. Nifurtimox is used in conjunction for more effective treatment and causes oxidative stress and cellular damage. Finally, we'll touch on the drug resistance. T. brucei has become resistant to diamidines by utilizing changes in aquaglyceroporins through mutations, deletions, and chimeric rearrangements. Eflornithine resistance involves alterations in the amino acid transporter, which affects DFMO uptake, DFMO being the irreversible inhibitor of ODC. Nifurtimox resistance occurs via type 1 nitroreductase, a major catalyzer of drug activation. And these are my references.